Hey guys, it's Joe coming from the ceiling speaker right now. Mo and I are going to teach you guys a little bit about protein metabolism. So a little intro into protein metabolism. Proteins, they basically are synthesized in the cell, and then wherever the cell needs it, it's transported to that cell, and then it can also be degraded if it's no longer needed. Synthesis of proteins is one of the most challenging biosynthetic processes, and it accounts for approximately 90% of the chemical energy used by a cell for a biosynthetic reaction. Now, kinda to get us a little started, we'll introduce to you guys the portion of translation towards the beginning, and then Mo will add in more details later on. So, in order to start talking about the genetic code, I'm going to start with tRNA. So essentially, tRNA, or transfer RNA, takes a amino acid to the ribosome so that it can form the polypeptide chain that has been used or that is templated by the mRNA, and those two will coordinate, and therefore you will generate your polypeptide. However, in order to get the amino acid onto the tRNA, you go through the aminoacyl tRNA synthetase, which will allow the amino acid to attach to the tRNA, and then you get a charged tRNA. Now, the goal was to generate, we know that each amino acid corresponds to a triplet. Now, these triplets are made up of 3 nitrogenous bases, and these correspond to their specific amino acid. Now we have 20 amino acids, so they were figuring out, okay, how can we take these 4 types of nitrogenous bases and generate 20 amino acids from those possible combinations. So as you can see, we have these calculations here, and what they found is they approximated there would be 64 possible combinations, which would account for the 20 amino acids. However, what they found was there was actually 61 because you had an overlap for the stop codons. So we have 3 stop codons. And again, a codon is just a triplet. So moving on, the genetic code here describes a process that you can refer to in your book, but essentially it talks about how they determined the genetic code and how triplets correspond to a specific amino acid. So in this case, they used poly U and found that it encoded for phenylalanine. So here up in the top right hand corner we have the genetic code, and this is a series of the codons or the triplets that correspond to their specific amino acid. Now some of these amino acids have multiple codons, and some of them do not, like methionine. You can see here AUG only has 1. And also the same with tryptophan. So tryptophan only has 1 codon that it corresponds with. However, there are 3 stop codons, you can see, and then the remaining others. So the anticodon is part of the tRNA, and I will show you a picture of that later on. One of the interesting things about this succession of the mRNA and the tRNA is that it's in a non-overlapping fashion. So basically what that means is it's just successive codons, so one after another, triplet after triplet after triplet. There's no overlapping, which would cause issues with amino acid formation. And you can look in your book at figure 27-3 to look at that a little bit more specifically. So as a result, we know that the reading frame, this just the linear sequence of the triplets, so like I was saying, non-overlapping, it's successive, continuous. This is what defines our proteins. Now there are certain molecular machinery that actually reads the mRNA strand, and if the reading frame is off by 1 or 2 bases, then the nucleotide is skipped, and then you get issues with the wrong amino acid being formed. So if you look down here in this figure, we see that in the original sequence looked like we wanted histamine, and instead we got a proline. So we can see how the rearrangement of even a single nucleotide can affect the overall polypeptide chain. Now, looking at different types and labeling them, a point mutation, as can be seen here, is a form of missense mutation, and this causes a shift in the reading frame. Missense is actually either an addition or deletion of the mRNA sequence. Now, the initiation codon is coded as AUG, adenine, uracil, and guanine. This codes for methionine, and then we also have termination codons, which are here. Now, these termination codons are also referred to as nonsense codons, which you may have heard before. And if we get codons of 50 or more, these are generally referred to as open reading frames, and typically non-genic DNA. Another vocab word that is often used in reference to codons and their respective amino acids is degenerate. Now, basically what that means is that some of the codons have, or some amino acids correspond to several codons, whereas other ones only correspond to one, as can be seen in these examples. Here's a little picture of my dad yelling at me when I was little. It's a mnemonic for stop codons. You go away, UGA, you are away, UAA, and you are gone, UAG. I know I look a little bit mad there, but it was probably for my own good. So moving on, so we're looking at the tRNA, and in this picture you can see the anticodon on the tRNA versus the codon on the mRNA. You see that the mRNA is in the 5' to 3', whereas the tRNA is anti-parallel and is in the 5' to 3' going opposite direction. So Watson and Crick hypothesized this phenomenon called wobble. Essentially what it is the first 2 bases of the RNA codon, these are always going to be your straightforward guanine, cytosine, adenine to uracil. These are your complementary base pairs, and it's going to be like that every time. However, the third base pair can be different in that it might not always be complementary. And so that's essentially what this phenomenon is referring to. and known as the wobble hypothesis. Previously, I talked about how there can be mutations in the sequences, and there are certain mutations such as missense mutations, as I talked about before, which are point mutations. These can be like additions, insertions, deletions. However, there are also silent mutations, and these play a big role. So silent mutations can basically, you can change a nucleotide or two nucleotides and still end up with the same amino acid despite these mutations that occur, because you have several codons that correspond to the same amino acid. Some amino acids only have one type of codon, however, there are many with multiple. So this can kind of help mutation resistance. Here also we have a transition mutation, which is just another type of mutation that can occur, and that's transferring a purine with a purine or pyrimidine, replacing a pyrimidine. Another key fact of the genetic code is that it's universal. There are some exceptions to that, but it's generally universal in all species. So next we want to talk about RNA editing. So essentially RNA or mRNA can be edited before translation via deletion, addition, or alteration of nucleotides. So the alteration of nucleotides can occur by the ADAR enzyme. Essentially, it deaminates the adenosine and turns it into inosine. And you can look into more in your book for the sake of time, but the defects in ADAR function, they can lead to human neurological conditions as is listed below. I'm going to finish up with talking about the protein targeting and degradation. I'm going to make this kind of short, but basically after post-translation, or after translation, where we have to ask the question, where do proteins go? So they can either be secreted, integrated into the cell membrane, or included into the lysosome. So where does this start? It starts in the endoplasmic reticulum. So there it will be modified and moved to the Golgi complex. And from the Golgi complex, it can be sorted and then sent to lysozymes or the plasma membrane or vesicles. And then eventually proteins can be degraded by proteolytic systems. Ubiquitin can tag the proteins, and then proteasomes can degrade the protein. And we're going to have this transition go to Mo to talk to you guys about translation and the specifics. But here's a little joke. And I wish I was adenine because I could get paired with you. And did you just mutate for a stop codon? Because you're talking nonsense. Alright, that's all I have for you guys. See you guys next time.